Howdy! We are Axil BioTech, and we're creating a plant positioning system to reduce current labor requirements for Raman spectroscopy. Axil BioTech is comprised of Marco Gonzalez, Connor Farrell, Matthew Cortez, and me, Bailey Voigt. This project is sponsored and supported by Dr. Kevin Ong, Dr. Dmitry Kurowski, and Dr. Rainer Fink. Global food security is an important topic today in maintaining our civilization. In fact, to maintain our current population and rate of growth, we're going to need 70% more food by 2050. Plant pathogens make a very large impact on crop productivity and cause 50% of all crop loss and cost us billions of dollars every year. Because this is such a detrimental issue, timely disease diagnostics are extremely important in increasing crop yield and reducing the cost of pathogen treatment. There are a lot of current ways to detect plant pathogens, such as hyperspectral imaging or thermography. You could also use more molecular techniques like PCR, immunofluorescence, or ELISA. These are all great methods of detection. However, each comes with its own drawbacks. From being extremely complex and requiring a lot of preparation to requiring expensive reagents that are not always readily available, plus a lot of time and energy. Our project works with Raman spectroscopy. This is a cutting-edge, non-destructive, and non-invasive technique that can provide valuable information on the chemical structure of what it's measuring. This technique has been used recently for both plant and human disease detection and was found to be extremely accurate. It has been able to distinguish between healthy and infected kernels with 100% accuracy, plus rose-rosette disease, among many other studies. This seems to be one of the best methods currently and is quickly becoming a strong competitor against techniques like PCR. However, it has its limitations as well, which we plan to fix with our device. Presently, human error can occur in the detection process because the spectrometer must be placed at a very precise angle and come in contact with a specific portion of the leaf, plus be held there for a correct amount of time. Several measurements must also be taken for the data to be sufficient. So, while very cutting-edge, current time and labor requirements for repeated measurements are hindering new discoveries in the field. Our system will interface with the current handheld Raman spectrometer and be capable of manipulating the position of a plant such that the leaves are positioned suitably for taking accurate measurements. Lastly, our project will be capable of reducing current labor requirements for taking repeated Raman spectroscopy measurements.
Hi. Now we're going to talk about the computer vision targeting assistance portion of the project. This will use an industrial camera with a 3.1 megapixel Sony CMOS sensor and the OpenCV machine vision framework. Using this, we will program an object tracking algorithm in C++ to identify and distinguish both the spectrometer and plant leaves from foreign objects. The computer vision targeting assistance system will communicate with the motion control system to reposition the plant such that Raman spectroscopy measurements may be taken. Now, let's talk about the motion control subsystem.
The heart of our plant positioning system is the motion control system consisting of the Atmega 1284P microcontroller, multiple closed-loop stepper motors, and a position feedback sensor network. Our product draws inspiration from CNC machining equipment, building upon those fundamentals to create the motion control subsystem of the plant positioning system. The motion control subsystem will allow a sample plant to be positioned so Raman spectroscopy measurements may be taken accurately and quickly. Now let's talk about the manual operator controls.
Let's take a look at our manual operator controls. These controls will allow for an operator to control all functions of the plant positioning system should they wish to operate everything manually, or adjust the plant after allowing the computer vision targeting assistance system to do the heavy lifting. These controls have a pair of joysticks, speed controls, and position hold switches for each axis, along with controls for the plant holding mechanism and the spectrometer. Now, you might be asking how all of these pieces work together. Don't worry, we'll explain that. First, we have the motion control subsystem. This holds and manipulates the plant. As you can see, the handheld spectrometer and the computer vision camera are both mounted to the motion control subsystem. Next, we'll add the remainder of the computer vision targeting assistance system to the picture. The computer vision targeting assistance system captures images from the camera, processes them, and sends commands to the motion control subsystem. The motion control subsystem then moves in response to these commands. Now, we'll add the manual operator controls. The manual operator controls read the values for the various joysticks, knobs, and switches, process the data, and send commands to the motion control subsystem. Now, we've talked about the problem and how we plan to solve it, but why should you care? Let's talk about that.
Once complete, our system will reside in the Plant Pathology and Microbiology building right here at Texas A&M University. From there, it will be used daily in cutting-edge research involving disease detection, where it can make great strides that in the end will help advance many fields for years to come. Thank you all so much for watching. If you would like to learn more, please check us out online at axilbiotech.com or feel free to email us. Our address is hello@axilbiotech.com. Thanks, and gig 'em.